Okay, so two basic kinds of microscopes. We have the optical and non-optical. Okay, optical means you have a direct image that is not processed by some computer. Okay, so you're seeing it, you know, it's with your eyes. So a couple different kinds. So we've got simple and compound. This is the simple microscope. Okay, what do you all know this as? Magnifying glass, but it is a simple microscope because it has one lens. Okay, so if you see this on a quiz or a practical, this is a simple what? Microscope. This is a simple microscope. All right. This is a compound microscope. Okay, because it has multiple lenses, two or more. Okay. All right. So you have the lens you see through the eyepiece, and you have a lens at the objective. Okay. We'll get into the parts in just a moment. You do need to know the parts of a microscope. I'm just going to tell you that now for the quiz. All right. So on the non-optical microscope, you have an indirect image. It's processed by a computer. Okay. So there's more technology inside of a non-optical microscope. You have, again, a couple different examples of a non-optical microscope. You have the scanning electron microscope. Think of the S in scanning for the S in surface. Okay, so you're seeing things on the surface of something. Okay, and I don't know if you can see these pictures down here. All right. You're getting the outside. Okay, so you're seeing the surface of all of these structures. Okay, that's what a scanning electron microscope does for you. Okay, you get all those cool pictures. All right. Now, other kind of electron microscope or non-optical microscope is a transmission electron microscope. So the scanning tells you the external features or the surface features. Transmission electron microscope tells you the internal features. Okay, so now instead of seeing this three-dimensional image, you're seeing what's on the inside. Okay, and it's more two-dimensional, right? It looks flat. Okay, so here you can see all these different structures inside of a cell. And when we get into covering cells in a week or so, we'll go over what each of these are. Okay, but the main thing is knowing the difference between a scanning electron microscope and a transmission electron microscope. Okay, so all right. So what type of microscope has a image that is not processed by a computer? Optical, thank you. What are the two different kinds of optical microscopes? Simple and compound. What's the difference? Simple has one lens, and a compound microscope has more than one lens. Right? It's multiple lenses, two or more. Okay, so if the microscope has an image that's indirect and processed by a computer, it is a non-optical microscope. Two different kinds of non-optical microscopes are scanning electron microscope and transmission electron microscope. Which one allows you to see the internal structures? Transmission electron microscope. Which one allows you to see the external or surface features? Scanning electron microscope. Okay, is that starting to make sense a little bit? Okay, you're gonna have to practice it, you know, but hopefully you kind of get the big idea. All right, so let's talk about, now that we've talked about types of microscopes, let's talk about the parts of microscopes. So you have different parts to a microscope, okay? So you've got an eyepiece or an ocular. It's what you look through, okay? Magnifies your image. You have a body tube, it's going to transfer light. And so I'll tell you, you're going to have to identify some, on the quiz anyway, you'll basically see a picture of a microscope. You might have to, let's just say there's a matching. I'll give you that much hint, okay? You might have to identify different parts and say this A is, you know, this one is A and this one's B, you know, it'll say these names here. I could also give you some functions in there and you have to identify, match the location with its function. Okay, so you need to know both. On a practical, I might ask you to write down the name of the structure, or I might ask you to write down the function of that structure. If I ask you to write the name, give me the name. If I ask you to write the function, don't give me the name, give me the function. Does that make sense? Yes or no? Yeah, yes, no, maybe, okay. So, all right. So the nose piece spins the objects around. So, all right. And you're supposed to have two hands on the microscope, so I'll show you that. But basically, here's your nose piece. Spins your objectives around, essentially. All right. You have the objectives. These are the different objectives. They magnify the image. Okay? They each have a different level of magnification. You have a condenser down here that focuses the light. Okay? So it's hard to really see, and we'll show you a better picture, but you can adjust this to focus the light. Okay? And diagram also allows you to adjust the light. Okay? The lamp down here is what's providing the light. Okay? And the base is your support. Okay? But wait, there's more, right? So this is your arm. So I feel like I'm doing my infomercial up here. Like you buy the new microscope, right? So you got it does this, it does that. Okay. All right. So this is the arm. Also is providing support for the microscope. This is your stage, right? Okay, it supports your slide. Move this, put the slide in. So you have the stage controls. You can move the slide around so you can adjust stage clips right here. Secure the slide. Then you've got these knobs here. This big knob is your coarse adjustment knob. Okay, it's coarse, focuses the image. The small knob is your fine adjustment knob. Okay, it gives you more fine adjustment. Okay, so when you're using a microscope, you start with the coarse to get generally where you can see it. Then you use the fine coarse knob or fine adjustment knob to get it down to where you can really see it really clearly. Okay, so and you can see that just from moving it. The coarse moves a lot when you turn it. Like if you turn it once all the way around, it moves a lot. Fine doesn't move that much. Okay, it's for a real specific, you know, movement, trying to really get to focus. You also have a rheostat, also helps to adjust your light. All right, so here's your picture. This is in your canvas. Okay, if you want to take a picture of this, come up here and take a picture, that's fine. But this is, I pulled these notes from Canvas, okay? You have access to the same slide then. All right, so again, here's your ocular, okay, eyepiece, your head or body tube, your arm, okay, your objectives, nose piece, your stage clips, here's your condenser, the diaphragm, okay. So the condenser is just above the diaphragm. All right, your lamp, your base, coarse adjustment knob, fine adjustment knob. Okay. So Those are your basic parts of a microscope. All right. So just to make sure you kind of have a sense of it, okay, what's this thing here? Anybody remember? Stage, good. Okay, what's this? Nose piece, right? Just, what are these? Objectives. What's this? Eyepiece, okay. What's this? Arm. This whole area here is a body tube, okay. What about this thing right here? Coarse adjustment knob. How about this one? Fine adjustment knob. The lamp, okay, very good. All right, so I'm gonna wiggle this piece right here. Can anybody see that? This is stage clips, right? And then the slide or stage adjustment knobs down here, just give you a better view. Okay, move the slide around. All right. So down here, can anybody see this right here, down here? You said it? No. So yes, the diaphragm, and then up here is the condenser. Okay. All right. Y'all got it. Y'all got it. All right. Care of microscopes. Always carry the microscope with one hand. No, 2 hands. Okay. I don't want to see any of this. I don't want to see this. Okay. You got to be like this. All right. So how many hands we use with the microscope? 2. All right. So when you're trying to unplug the microscope or unplug the plug, pull the actual plug here at the outlet. Don't just yank the cord, okay? You can damage the cord that way. Always put the stage all the way down, okay? So make sure that your stage is all the way down when you set it up. All right, so you got to make sure. So like we can adjust the stage, make sure it goes, put it down. All right. Mechanical stage should go in the middle. Okay, so that's this stuff. So you want to make sure it's not hanging out on the side because this can get damaged. All right. I don't know if y'all can see that there, but we want to make sure we get that all the way in. Okay. Scope should be returned to the scanning objective, and we'll show you which one that is in just a minute. No hanging cords. So you see this set up like this, and I ask you, all right, is this microscope stowed correctly? What would your answer be? No, why? Cords just hanging out, okay? So some of them have little hooks around here where you can kind of do like you do with the vacuum cleaner and stow it like that. Otherwise, you got to have it wrapped around. Okay. So no hanging cords. All right. Some definitions you need to know. Okay. Parfocal. So it just means if you're changing the objectives, if we're moving from one objective to the next, image remains in good focus. So keyword there is focal. All right. To be distinguished from Parcenter. Keyword here is center. As you change objectives, the object remains in this center. Okay. So if you're moving it around, you don't want the image to slide to the left or to the right. You want to stay in the center. That's Parcenter. You change objectives, you want it to stay in focus. It's Parfocal. Now the field is what you can see. The area is visible through the microscope. That's your field, what you can see. Okay. Anything you can't see is outside the field. Okay. Depth of field means depth or thickness in the specimen. Okay, depending on how it's cut, how thick it is, you might see more depth or less depth. Okay, that's simultaneously in focus. So depending on what kind of microscope you have, you might be able to see lots of depth of field, you might not be able to see. And I'll just tell you, with this kind of microscope, you're not going to see a lot of depth of field. Okay, those scanning electron microscopes we showed you before, showing the surface, you saw lots of depth of field, right? You can see it's three-dimensional. Not so much with these. Okay, so parfocal, parcenter, field, depth of field. All right. So review those. The area you can see in the microscope is called what? What do we call that? The area, the visible area. The field, right? Okay. As you change objectives, the area remains in the center. What do we call that? Parcenter. Being able to see some depth or thickness in your field, we call that depth of field. If you change the objectives, the object remains in relatively good focus, we say that's okay. And we'll just make sure we remember the area you can see in the microscopes. When you're looking down at the microscope, we call that the field. Okay. All right. Total magnification. Total magnification, I can just tell you, for the practical, you're probably going to have to do this. You may have to do it on the quiz. I can't remember, but I know you at least will have to do this on the practical. Total magnification. Okay, and this is real super fancy calculation. Okay, you take two numbers and you multiply. All right, that's about as fancy as we're getting today. Okay. All right. So you take the magnification of the eyepiece, you multiply it by the magnification of the objective. And I'll just tell you, for the purposes of the exam, read the question to find out what this is. Okay, don't just assume. If it tells you what it is, use that number. Okay, so you multiply it times the objective magnification. Now here's those different objectives. Okay, so most of yours are going to have 3 or 4. And most will have 4, you might have a few in here that just have 3. So lowest magnification, lowest power is scanning. So remember we said we want it to be, the microscope to be stored at the scanning objective. Okay, so that's at the 4x. Okay, the shortest, and the color associated with that is red. So I don't know if you all can see that, but the numbers there are red. Okay, and it's the shortest one, shortest objective. Now a little bit longer, you get the low power, yellow. Okay, so we'll turn that, the letters are yellow. This is 10x. A little bit longer, you get to high power. It's blue, the letters are blue, color's blue on it. It's 40x. And fourth one is oil immersion. Its colors are white. It's the longest one, longest objective, 100x magnification. All right, so how does that relate to total magnification? Well, I'm glad you asked. So let's just say, for the sake of this example, we're going to go with an eyepiece magnification of 10x. And again, like I said, read the question. Maybe it's 10x, maybe it's not. Okay, but you got to figure that out. So the eyepiece magnification is 10x. For the scanning electron microscope, what's the total magnification then? 40x. Okay, when we don't do x squared, we just say 40x. Okay, so, and that is basically 10x times 4x. All right, so again, we don't do x squared. You know, I know this is not technically college algebra, but it's 40x. All right, Low power objectives, 10x. What would the total magnification be? 100x. Okay, so how did I get that? 10x times 10x, right? Okay. High power, 40x is the objective. So what's the total magnification? Okay. How did I get 400x? 10x times 40x. And oil immersion. What would it be? 1000x. All right. Y'all see how that works? Yes, no, maybe. Okay. Now I'm going to change it up on you because, like I said, you got to read the question. What if the eyepiece objective is 5x? What do we do now? What needs to change in this equation? That's what changes, right? You just, instead of multiplying by 10x, multiply by 5x. Now what do we get for a total magnification for our scanning objective? 20x. For low power, 50x. For high power, 200x. And for oil immersion, 500x. 5 times 100. Okay, so just want to make sure you know just how the math works on that. Okay, so yeah, I think y'all pretty much got that. So all right. So just to review, what are our 4 objectives? What are their names? Start with the lowest. Scanning, and then low, and then high, and then oil. What is the objective, or what is the magnification of the scanning objective? 4x. What is the color associated with 4x? Red. All right. What is the magnification for the low power objective? 10x. What color is associated with that? Yellow. Okay. For the high power objective, what is its magnification? 40x. Okay. And what color is associated with that? Blue. Okay. And then for the oil immersion lens, what is its magnification? 100x. And what color is associated with it? White. Okay. All right. So y'all know what you need to know. All right. So what I want you all to do now is look under, basically in the middle. Some of you have microscopes, so if y'all can pull them out if you have them. All right. Use your 2 hands, just like we learned how to do. You got 2, pull both out. Yeah, some of them have 2 lenses, 2 eyepieces, some have 1. It's okay. So what I want y'all to do, send someone up, come up here and get a colored thread slide and a letter E slide. And I want y'all to practice trying to focus. Okay, again, start with the scanning objective. Use your coarse objective to find it, and then use your high power. And then you can start changing objectives. One, so there are going to be 2 slides. So maybe one can have one and one can have the other. All right.